Sean McVeigh, Training Coordinator at the National Soil Survey Center, and welcome you to today's webinar by Dr. Paul McDaniel on properties and spatial distribution of Spodosols in the Northern Rocky Mountains. I do have a different teleconference line today, so I'm going to ask that everyone except Paul and I mute your phones because I don't have Paul set up to join me as a presenter link. And so every extra comment people make or desk drawer that they open or throat you clear and cough and sniffle, we hear all that and it interferes with the recording and takes away from the webinar. So if you would, just please, everyone but Paul, mute your phones. We're using live meeting today, and as I said, the presentation is going to be recorded. You can ask a question. We can open it up at the end for audio questions, or you can simply type in your message at the Q&A tab at the top of the live meeting frame, and we'll take the questions at the end. With that, I'll go ahead and turn it over to Paul and start the presentation.
Okay, thank you, Sean. I'd like to maybe begin by mentioning the word Spodosol. Now, I know a lot of people say Spotosol, and I say Spodosol, and some others. There's quite a bit of disagreement about this. So, for those of you that say Spotosol, I will apologize now. And wherever I say Spodosol, you can insert Spotosol. And I'd also like to acknowledge the people who've been very closely involved on this project. Mitch Valerio was a graduate student here and now is with the Ohio Department of Natural Resources. Paul Getzler, soil scientist and spatial analyst here, and Bruce Knapp was recently retired from NRCS, but was the MLRA head here at Podzolization. Sean?
Yeah.
To advance time,
There's the arrows in the lower left corner.
oh, right, okay. There we go. Okay, let me begin by saying a little bit about the study area. Part of MLRA 43A, which is the Northern Rocky Mountains. You can see on the map there that MLRA 43A is situated up on the Canadian border. And what we're really going to be focusing on today for the purposes of this talk is an area that is encompassed by the Canixo National Forest. You see the area of the National Forest there in northern Idaho going up to the Canadian border, spilling over to Washington. And that's where this study will focus. A little bit about the study area. It's a glaciated mountainous landscape, as you can see in the photos, and interestingly to me, anyway, is that much of the middle to high elevation part of this landscape is mantled with volcanic ash, and I'll say a little bit more about this in a second. We have quite a range in elevation, from about 650 up to 2,300 meters in elevation. The soil moisture regimes range from xeric, lower elevations, to nudic, and higher elevations. Probably representing a range in mean annual precipitation from 700 up to 2,000 millimeters. And soil temperature regimes are frigid and dry in the area. Volcanic ash influence. I think most of you have probably heard of the Mount Mazama eruption, and you see the graphic on the left here with the Mount Mazama shown down here in southwestern Oregon, erupted about 7,600 years ago. This area that you see, the blue shading, represents the large ash fall from that eruption. And if you look at the image over here on the right, this is a soil profile from up in the study area. And what I wanted to point out is this entire So even though it's a fairly, very thin layer of ash, it doesn't contribute a lot in terms of parent material, but it's a useful indicator of stability. In other words, where we find this real thin layer of ash, we know that there has not been significant disturbance in the low profile for the last 2 years.
Just a minute, Paul. Paul, just a minute. We're getting a lot of interference, so just to remind everybody, please mute your phone. And if you don't have a mute button, *6 will mute your phones. Thanks.
So back in the 1980s, there was some work done up in the study area, focused in an area known as Trout Creek, which you can see on the left-hand side of the image here. And what was found during the course of some investigations up there is that we looked at soils that are at middle elevation. So you can see the profile here in the upper right. You notice this grayish or whitish layer up here. And as we continue up in elevation towards the lower left-hand corner of the image here, you see this soil profile that has a very distinct gray or white-looking layer. So based on some of these field observations, laboratory work that was done, it was recognized that we actually have podzolized soils, and that these podzolized soils kind of formed a gradient that coincided with the elevation gradient up there. This brings up the subject of podzolization, and I wanted to spend just a minute or two with some of the basics of this. That podzolization is simply the chemical migration of aluminum and iron and organic matter, or I should say and or organic matter. And usually this migration is all three of these components moving together in the soil. This example of how this might work is shown here in this little schematic. We've got an aluminum ion. Remember, aluminum is a +3 ion, which most forms that you find in soil tend to be relatively mobile. But if you take some organic, in this case, very simple organic acid, oxalate, the oxalate molecules can chelate the aluminum and render it much more mobile. And the net result is that we see this translocation from the heat rise down into a DHS or HPS rise in some cases. And in terms of soil taxonomy, it's this process of podzolization that gives rise to albic, spodic, diagnostic horizons. And in terms of the taxonomy, this would give us Spodosols, and in some cases, depending on how well expressed this podzolization process is, we might have spodic or integrated. What this looks like chemically, you can see illustrated here in this graph on the left. What I've plotted here is the function of depth. In red is the oxalate extractable iron, and you can see the bulge, the depletion here in the heat horizon, the bulge in the DHS horizon, and the blue curve here is for aluminum, and very similar trend. And then you'll notice the organic carbon distribution step, where it's relatively low in the E, bulge here in the DHS, and you see the decrease in depth. So this is what this podzolization process looks like chemically. And if you think about some of the environmental conditions that are going to favor podzolization, high precipitation is certainly one of the main factors, and as a result, we tend to see an awful lot of Spodosols in more humid areas near precipitation, and especially those that have a relatively high snowpack, because the snow tends to melt very slowly for a long period of time, and it provides a lot of water to cool down through the soil to drive this podzolization process. Cool temperatures also are associated with completing Spodosols, and it's thought that these cool temperatures tend to inhibit the decomposition of some of these simple organic compounds that are probably involved in the podzolization process. Coarse textured parent materials also help promote podzolization. It's a lot easier to move soil solution through a coarse textured material with less pore space than it is through a finer textured parent material. And then, for instance, irrigation, vegetation, tend to promote podzolization as well. And in the study area, we certainly have a lot of coniferous forests, and these coniferous forests seem to provide the right sorts of organic materials to supply chelate to drive podzolization. And I must mention this last item here. Forget these first four things, but then you've got poorly drained landscapes and humid regions, such as the southeastern United States. And I think in these cases, the spodosols that are found there are different in terms of their genesis. They form more from the bottom-up type process. This used to be called groundwater potholes, and it's clear that the groundwater, the rise and fall of groundwater, and some of the dissolved constituents plays a big part in the formation of these. But what I'm going to be talking about today is really focusing on these first four things up here, because these are the sorts of environmental conditions that we have. One last thing I want to mention about podzolization. A lot of times I have been asked, well, so what? Podzolize, spodosol, what's the big deal? Well, I think in order to answer that, we need to look at a couple of chemical characteristics of podzolized soils or spodosols. And I have two examples here. On the left is a soil from northern Idaho. It was part of the study. On the right, state soil in Maine. What I included below is some of the lab data. And I'm just going to focus on, first of all, pH. And one of the things about podzolization gives rise to very strongly acidic soil horizons. You can see, especially in the Maine soil, this pH is down in the E horizon around 4 and going up to 4.7, a little bit higher in the Idaho soil, but still very acidic. And some of the podzolized soils that we have looked at from the study area, we have found pHs in the E horizon, and the pHs in water as low as 3.5. Very strong. And the other thing you might notice in the table, the extractable aluminum. You can see in both of these soils the quantity of extractable aluminum, which is roughly equivalent to exchangeable aluminum, is quite high. And because of this, we end up with aluminum saturation percentage. You can see there it's upper 80s and 90s. So these are very aluminum-rich soils and lots of aluminum activity. And because of the detrimental effects of aluminum for most biological systems, I think it makes fairly clear that the chemistry of these soils is going to have a big effect on biological processes, whether it be tree growth, nutrient cycling, and just the whole gamut of these biologically mediated processes. So I think that really is the reason for the importance of these soils and why we definitely need to recognize them. Okay, I want to kind of go through podzolization now and talk a little bit about how this project came about and talk about the results. So the work that I mentioned that was done back in the 1980s was admittedly of very limited geographical extent. And you can see the area up here is essentially drainages in the Selkirk Mountains of Idaho. So even though we were aware that these soils were present, we had no idea how extensive they were in other areas. Now, given that, the fact that if you look at a map of the available soil survey data for the United States, I'm sure you've all seen this type of map, you notice that the western United States looks quite a bit different than the Midwest and eastern United States, in that we have a lot of unshaded areas out in here that, for the most part, represent federal lands. And in fact, if you zoom in up here to northern Idaho and look at the areas of what constitutes the study area, you can see the predictive national forest up there. And this is part of the area that does not show up in its So there's been an effort underway to take some of the forest land and existing soil data that they have, soil survey data that is present, and get that converted into a form that is consistent with National Soil Survey standards, so that these areas can be converted or included in maps and web soil survey. some of you might be familiar with what's known as the land system inventory. And up in the study area, this was done back in 1970. And I mentioned currently trying to get this information incorporated in NASA's web soil survey. But there were really two main issues. One is that most of this mapping, the LSI, occurred prior to the establishment of the ANSI. And as it turns out, that's not too big of a problem because many of these volcanic ash influenced soils that are now Neanderthal were classified at the time as ANSI. So it's really just a matter of translating the older and newer taxonomy. I think what is maybe the larger issue here is the second item, and that is that the Spodosols were not recognized for various reasons in this inventory. I don't know exactly why that is, but nevertheless, we know these soils are there, and I think it's important in the interest of including the most up-to-date and complete information in the soil survey that are recognized. So this kind of served as the empathy for this project that I'm going to describe. And the idea was we wanted to try and develop some sort of a terrain-based model up in the study area that we could use to predict the spatial distribution of the soil. And then secondly, try to produce a digital map layer that we could take and supplement existing soil survey information. So that's really what drove this whole project. And I'm going to talk a little bit now about how we went about this. And so I'll begin with the method. And in order to do this project, one of the things that we're up against is that this Nixon National Forest represents an area of 1.6 million acres. And obviously, that's a very large area, and much of that area has very limited access. So what we wanted to do was come up with a way that we could go out and sample that area in a meaningful manner that would allow us to make some pretty strong predictions or develop a strong prediction model that would serve us well for the purposes of identifying areas where these soils are going to be. Now, based on the previous work that had been done up in the area, we knew that elevation was definitely a driving factor in where we could find Spodosols would be at higher elevation. And the other thing that we recognized was solar insulation, or if you want to think of that as an aspect. In other words, when you get to the high latitudes, when the slopes are facing north or south, so with this type of stratified random sampling, what we did is we took the entire study area of 1.6 million acres, and we divided it up into 9 different categories based on low, medium, and high elevation, and low, medium, and high solar insolation. And a high solar insolation would represent a slope that is facing south. Low solar insolation would be, think of that as slopes that are primarily northerly. So we end up with these 9 categories, and we selected in the study area, we broke it into these 9 different samples. And what we did was, within each of the combinations of elevation and solar insolation, we would randomly select, I think, about 15 or 20 locations across the study area. So you can see the purple color represents low solar insolation, low elevation, and these little purple areas here. By doing this, what we did was make sure that we had a very good sampling of the range and variability that we find in these environmental factors. And I'm going to zoom in here on the area that you see there. So this is the Trout Creek area. You can see how the landscape is broken up into, down here, this brown color representing high elevation, high solar insolation. Here's the other side of the ridge, and you've got the lower solar insolation, but high elevation. So this was kind of the sampling strategy. So once we had this set up, see them shown here in the red circle, scattered around the study area. And what this represented is 8 sampling sites from each of the combination of solar insolation applications. So it gives us the total of 72 sites. The next step then was to determine, based on what we saw in the field and also based on some of the laboratory results, we had to determine soils present in each site. And we ended up with really 4 different types of situations. And I'll illustrate those here. You can see, first of all, the image here on the left. This is a good spotosol that meets all the taxonomic requirements in terms of morphology and also chemistry. We also ran into soils, like you see in the profile here on the right, where they have T and DHF, DF type of morphology, but because of, in most cases, the color of the heat horizon being not quite light enough, maybe one color chip off, and even though they're podzolized soils and they have the DHF horizon morphology, they technically don't qualify as Spodosols, at least the way soil taxonomy is. We also found soils, like you can see here, that would have a spodic horizon, and probably this zone right down in here in the soil profile, where we don't have a, it meets the requirements for spodic horizon, but we don't have so. Finally, profile on the right, look at this, we did not see any kind of indication of spodic horizons for Spodosol morphology. So these were really the 4 situations we ran into. And what we decided to do, you can see the breakdown here. Out of the sample sites, we had 10 Spodosols, we had 16 that had the morphology, but as I mentioned, they lack exact color requirements. Then we had 17 with spodic horizon and then 25 with non-spodic category. And what we decided to do with these was to group the good Spodosols and the one-chip Spodosols together for purposes of this study, calling them the bottom 2 categories. They were spodic horizon. And a lot of times these just showed up as a result of the screen based on the lab. The non-spodic one, and we call them non. So you'll notice that the total here is only 68 samples instead of 72. What happened is we ran into a couple of sites that, and looking at the data, we realized the disturbance that they could get close to getting crossed out. So the next step then was to try and build a model. And as I mentioned earlier, we wanted to use terrain attributes, other types of environmental attributes. And you can see a list here of, it's not a complete list, but many of the different types of variables that we use to try and construct a model. Everything from elevation, slope gradient, latitude, longitude, bedrock geology, presence of particular plant species, of alpine fir, paragraphs, and different things related to aspect and radiation. So the approach that we use is to use something that is known as well, let me back up just a second and show you the results here. I think this is quite interesting. This figure here shows the soils that we classified as Spodosols, red, non-Spodosols, green, and you can see them arrayed on this figure. Now, the figure is set up so that up here at the top at 0 degrees, in other words, due north, over here we've got 90 degrees, which would be due east aspect, here 80 degrees or south, down here 270 west aspect over here. And then the other thing that is shown here is elevation. So imagine that this figure is like a cone coming out of the screen action. So this line, this circle represents elevation 500 meters, 1,000, 1,500, finally here in the center, an elevation of 2,000 meters. So you can see the array of these points as a function of aspect. general, the red does tend to cluster in the part of the graph here, with a couple of exceptions. Greens are a little bit more spread out, but by and large, we get Spodosols, this sort of an arrangement, and non-Spodosols here. So, to try and develop a model for this, we use classification trees. Try and walk you through how we did this. So we take all of our training data, which is basically the 68 site, whether it is Spodosol, and then we put this information into the computer, and the study area is divided up into 30-meter pixels. And for each of these pixels, we have a variety of terrain attributes assigned to the pixel, and also other environmental attributes. And all of this is put into a program. And what these classification trees do is they start to break down the data and figure out which sort of variables are able to separate out the two classes you're interested in, which in this case is non-Spodic and Spodic. So for example, we take all of our data, and the first break in this classification tree has to do with radiation, solar radiation. And so what this represents right here is high solar radiation. And when that's the case, we end up with, out of our training set of data, 20 of those soils were non-Spodosol, and 0 came out to be Spodosol. So then the remaining data is moved along this tree, and we come to elevation rate. You can see the elevation here is 1,132 meters. So the soils that are below this come down here, and out of those, we had 12 of them were non-Spodic, and only 2 were Spodic. The remaining data then moves down this direction. We've got another term in here for solar radiation. In this case, this is low solar radiation. And go down here. We don't really see, we see 6 non-Spodic, 4 Spodic soils, so kind of a mixed. And same thing when we get down to the final node tree, we're looking at elevation. This is elevation less than 1,500 meters. And we end up with 10 Spodic soils, 4 non-Spodic. And what that leaves us with, think of this as soils greater than 1,500 feet elevation, 10 Spodosols, and 0 non-Spodic. So this is how a classification tree establishes rules to predict where we're classifying the Spodosol. So what we did with this, and this turned out to be the best model that we could generate using different combinations of variables. So you can see that down here on this end with the red circle, we know that that's a pretty good predictor right there. That area represents low solar radiation, high elevation. Conversely, down on the other end, if we look at these first 2 nodes, we end up with the majority of the soils not showing solar photovoltaic, and these represent the areas of high solar radiation and lower elevation. So this is the kind of information we need to try and figure out how quickly this happens. So again, we take our study area and all these 30-meter grid cells that we have, and we apply the model. So for these first 2 nodes, our interpretation of that group of soils is that Spodosol essentially has what you don't expect to find at lower elevations and lower solar radiation, or excuse me, higher solar radiation. Then down on this end, we get to this final node here where Spodosols are dominant. These are the higher elevation soils with fairly low solar radiation. And then those are clearly where Spodosols are predicted to occur. Then we end up with this problematic movement here, where if you remember from the previous slide, these represent the kind of a mix of Spodosols and non-Spodosols. So really the best way we could come up with to handle this is to label these as Spodosol / integrate, such as the Spodic integrate. Spodosols may be present, and some of the Spodic integrates may be present as well. Well, So we take this information and then we apply it to our 30-meter grid cells. We're able to generate a map. And you can see the resulting map here. We've got the colors red, yellow, green. Red would be the soil where we expect soil to be. Soil predominantly be Spodosols, about 11% of the study area. The yellow, where we might expect to be Spodosol or a spodic integrate, about 12% of the area. Finally, the green area is represented about 77% of the study area. We don't think we're going to find Spodosol. And I guess maybe the point here is that we've got about 23% of the study area in this red and yellow color where we do see podzolization around. So next step then was to try and take our model or our map and vary this somehow to the land system inventory. And you can see in the upper left corner, this is a portion of our study area, and you can see the polygons with the mapping and investigation numbers. Down below is our model. Now remember, we're dealing with a polygon-based system here and a raster-based system here. So what we did was simply combine these two things, and just for a second, I don't see the color coming through on this for some reason. I'm going to go on to the next one. But what we decided to do was we used an algorithm where we looked at each of the polygons, and if the majority of the grid cells within that polygon were in our Spodosol category, then we assigned that entire polygon. Same with the yellows and also the green shading. So doing that, then we ended up with a polygon-based map with essentially the same interpretation that we had before. The red color being where we expect to find Spodosols, the yellow Spodosols, maybe spodic integrate, green, which is good. So the last thing that we wanted to do was some sort of accuracy test and see how well the map that we generated was going to work. So we had 33 more or less random inputs, and we went out with NRCS personnel and we tried to assess the morphology of each of those things and see whether or not that agreed with what was predicted from our map. I can show you the results right here in this accuracy matrix. So it's a little bit complicated with a lot of numbers here. These columns that you see right in here are what's predicted by our map that I showed you previously. These rows that you see here are labeled actual. This is what was determined based on what we saw in the field. And I guess what I want to point out is that in terms of how our map predicted things, we look first here at the non-Spodosols. You can see that there were a total of 14 of our assessment sites, and our map predicted 3 of them were going to be non-Spodosols, or 3 would be Spodosols, 11 not be. Let me back up here. Sorry. That we had 14 that our map predicted as non-Spodosols, but actually 3 of those turned out to be, and 11 turned out not to be. So overall, our accuracy for the non-Spodosols is about 78.6%, pretty good. For the Spodosols, not so good. In other words, you can see 52.6%. What that means is that our map tended to over-predict Spodosols. So we predicted there would be 19, but based on our accuracy assessment, only 10 of those 19 actually were Spodosols. So not quite as high there. This is our overall accuracy there, about 63.6%. And I think one point I'd like to make at this juncture is that the problem that we ran into is some of these areas on our map that show up in yellow. And remember that I mentioned that was the problematic group of soils. And based on what we could see in the field, we simply had to go on the basis of morphology. We did not subsequently take samples from lab work to determine whether we were in an area that showed up on our map as yellow, we were saying, well, we should see some indication of Spodosols there, and as it turned out, we really didn't. But still, I think the overall accuracy is fairly reasonable and probably could be improved upon a little bit. So, just in conclusion, a couple of points. And the first one is that effective precipitation and snowpack we know to be important in driving the podzolization process and elevation. All of these are proxies for effective precip. Solar radiation, throw that in there, and we can do a pretty good job of predicting spodic morphology. And the second point here is that the spodic morphology comprises a fairly significant portion of the high elevation landscapes within the study area. And given the extreme, the acid nature of these soils, the high amount of acid, high saturation, I think it's really important that the podzolizers recognize anyway. So I think with that, I'm going to go ahead and stop there and maybe open things up for questions.
All right, well, while people gather their thoughts for some questions for Paul, I'll open it up here in the room to some questions, have people come up and ask Paul a question. There are just a few rules, there's not very many people on the line, but I'd ask if we take turns and we could ask them orally, just so you can take your phone off of mute, and we'll open that up for questions. Anybody in the room here with me want to ask a question? All right, seeing none, I'll open it up to the rest of the audience.
Hi, Paul, this is Tom Burke in Alamosa. I was wondering, was there any correlation between frigid and chronic areas with Spodosols?
There is. In fact, if you look at where the frigid cryate breaks are placed in the study area, the cryate soils are the higher elevation soils, and certainly most of the Spodosols would have a cryate stream. And I guess maybe the other part of that I wanted to mention is one of the things that we're trying to do now is look a little bit at some of the snowpack information that's available from some satellite imagery. And we do see a pretty good relationship between where we find the Spodosols at higher elevation and where we tend to see the snowpack play in for longer periods of time. And definitely the cryate soils would encompass most of the areas where we can see podzolization well-expressed.
So the non-spodic soils that are integrated with it, are those mostly alpha sols, dipsols, endosols, or are they all mixed up?
Okay, I guess the short answer would be that they're all mixed up. Now, a lot of them are endosols. And the thing that we found when we got into looking at some of the specifications that the endosols would, in some cases, there would be a spodic intergrade recognized in the taxonomy. So that's certainly part of what we're looking at. In other cases, inceptosols would be some of the main soils that we find would be spodic horizon, and not so many alpha soils. We don't see a lot of that area, primarily because at least the areas with very coarse texture, granitic, bedrock geology, and generally we don't find alpha soils there. So I would say endosols and will probably be the dominant soil groups that find the spodic horizon.
Okay, any other questions? Okay, I've got a question here in the room from Sky Will. She's coming up so she can be heard.
Hi, Paul. I have a couple of questions for you. One is on your the stratification used for sampling, were those fairly equal in extent? Each of your 9 classes, do they have about the same area, or?
Yeah, that's a good question, and I guess I didn't mention that. But the way that we put them into the 9 classes was so that we would roughly have, in terms of elevation, 1/3 of the land areas at low elevation, 1/3 of the study areas at medium elevation, and 1/3 of the high elevation. And then we did the similar thing with the solar installation.
Okay, that makes sense. And the other thing actually kind of came up in the last question, but I was wondering if you thought about trying to predict intergrades, or maybe it's the population of all those spodosols is part of what's giving you some difficulty in the predictions.
Well, I think that, yeah, definitely the almost spodosols, the ones that are leaning that direction, is where we see the problem with the current model.
Yeah. So
the good spodosols, the ones that meet the letter of the law and taxonomy, and I would say even the majority of the spodosols that have the right morphology, that look like spodosols, but maybe miss the letter of the law by one color chip, those aren't really the problem. It's the ones where you've had podzolization to some extent, but it is not as well expressed. And a lot of times we found we couldn't recognize people.
Oh, okay. So you can recognize them in the field, and you don't have any remotely sensed information that tells you what those might be either.
No, and that's kind of the something.
Crux, yeah. Okay, thanks.
I've got a question online, Paul.
Okay.
And the question is, would it be helpful to allow feature-level attribution, percent composition by polygon, when combining the modeled raster results with the historic Forest Service vector mapping?
And I guess my answer would be yes. You know, we were really looking at this kind of a first cut, just to see if we could find some relationships between the Forest Service mapping and what we were able to predict on the basis of our model. But yeah, I think that definitely would be something that could be looked at and utilized in the future.
Any other questions from the audience? I've got one here in the room with me from Joe Shreddy. He's coming up so he can be heard.
Hi, Paul.
Hey, Joe.
Paul, of the, what percent of the spodosols had andexoid properties? Just roughly?
I don't know that I can give you an exact figure on that, Joe, but most of them did.
Yeah.
So where we found the spodosols, typically those were the good andic soils as well.
You know, you had communicated about these E horizons fail the Albert criteria because the value is just a little too low. So, you know, they're close to being spodosols, but they don't quite make it because they don't have the Albert horizon.
Right.
The soil taxonomy has the option that if the soil does not have andic soil properties, meaning that it doesn't have a strongly expressed spodic morphology, it can still be a spodosol if it doesn't have andic soil properties. And you did have some that I remember looking at that met that criteria, but if many of them in that ash mantle do have andic soil properties, then they would fail because essentially they would be better classified as andesols, which follow spodosols in the key to the orders. So it's
Right.
You know, the...
And,
go ahead.
well, and this was something that we really looked at in a lot of detail because we really wanted to get them into spodosols because they look like it, they've got the chemical characteristics, but you know, it's one color chip thing. But as I said, we weren't able to do it. Even though they don't meet the letter of the law
Thanks, Paul.
Any other questions from the audience? Looks like we have one online again. And Sharon's raising her hand. So Sharon, I don't know if you want to just ask your question if you have one.
Hi, this is Sharon, but it's really Tom's question. I'll let him ask it.
Yeah, I just couldn't remember the figure from your accuracy assessment. Did you include your intergrades as one of the classes, or was it just non-spodic and spodic?
Well,
And then so, if you threw in the intergrade as a class, how do you guys figure that that would have affected your final accuracy assessment?
well, if we threw in the intergrade as a class, I think it would have definitely helped our overall accuracy assessment. The problem being, as I mentioned earlier, that we found that we really were not able to consistently identify those intergrades in the field. Some cases we could, but there were others that we couldn't.
Right.
But no, I think that when we had to, since we were only using the two classes for the accuracy of the assessment, and we had the pre-map classes, it really created some difficulty. So I think that's the reason that we were over-predicting, the reason that our map was over-predicting the Spodosol distribution, because all those areas that showed up in yellow, we wanted to see some kind of spodic morphology. And we often did.
Okay.
Hey, Paul, this is Eva Muller in Bozeman.
Hi, Eva.
How you doing?
Good.
Yeah, I was wondering if you thought about the bedrock geology, like the granites might be more prone to form the spodosols with the coarse texture versus some of the other parent materials there. Did you consider making that one of your breaks?
Yeah, and again, that's a really good point, because when we got around to analyzing the data and the distribution of the spodosols, one of the things that we did notice is that there's definitely a difference, those soils on top of the granitics versus those that are on top of the finer grained metasediments in that area. And unfortunately, we weren't able to incorporate it into the overall study design, because we started off with elevation and some solar insulation and rocks in different aspects. And the more variables that you start to put in to your sampling scheme, the more classes that you generate. And then you either have to get more samples, or you have to have these classes where you have fewer samples in. And then you really start to lose the power of the classification tree approach. So it is something that we feel pretty strongly makes a difference. And yeah, if we had more time and more money or work on this, that would definitely be a component that we want to look at.
Any other questions for Paul? All right, hearing none. Paul, I thank you for the webinar today, and this will conclude our presentation.